So we'll talk about what metabar coding is, some background, why the bioinformatics and some of the analysis are different from traditional barcoding, some research progress in metabar coding, a little bit about our development of bioinformatics tools to do metabar coding, and some looking into the future. And as always, you can stop me anytime for questions. This is okay. Okay, so barcoding is when you expect you have one specimen, mariposa roja, and one DNA sequence, okay? 1 to 1. So that's the main thing about barcoding, right? Anytime you get 2, it's non-target sequence or contamination. With metabarcoding, it's many to many. So you have many specimens or species in a sample, you extract the DNA together, you sequence together, and you want to see what's in there, right? So that is the main difference, okay? So let's just go over some basic questions about what we think we would use for different approaches, metabarcoding or barcoding. So if we wanted to build a DNA reference library, would we put your hand up if you think we would use barcoding and metabarcoding? Okay, we'll go through at the end. Okay, what about if you had a specimen and you want to say, I have this specimen, I want to know what it is. Would you use barcoding? Put your hand up. One specimen, or metabarcoding? Okay. What if you wanted to say, you want to see if a species is in an area and do that for very cheap? Would you use barcoding or metabarcoding? Okay. And if you wanted to, have you had a sample and you wanted to know how many individuals of each species were in a sample, would you barcode everything or metabarcode everything? Okay, so here are my answers. Oh wait, I guess I didn't put them in. Okay, so for the first one, building a reference library, maybe that slide is hidden. I think you would do barcoding, not metabarcoding, because you need to have the single sequence associated with each specimen to build up your library. For identifying a specimen, you would also do barcoding because you want to know one sequence for that specimen. To know if a species is in an area, that is what you would do metabarcoding, right? Because it's cheap and you don't have to process much. You just extract DNA and that's that. And to determine abundance of species in a sample, that one, I'd say there's no right answer, right? It's expensive to do it with barcoding, but there's some ways with metabarcoding to figure it out by maybe the number of reads of sequences that you get. But maybe you would do barcoding if you have a lot of money, if you don't have a lot of money, you would do metabarcoding. But really, metabarcoding, the main reason we use it is to determine what species are in a sample without spending a lot of money. That's the main reason you do metabarcoding, to save money. And yeah. I think there are 3 main challenges with metabarcoding that you don't get with barcoding, that you need to have bioinformatics strategies to deal with. First, as you said before, one sample, many sequences, okay? So, you need to figure out how to cluster the sequences in the single sample to understand what's there. Second, this is a somewhat unique problem to metabarcoding, although it can happen in barcoding. You get what's called chimeras, okay? Where you get the different DNA from different species that joins during PCR. So you get molecules that are real DNA molecules, but they don't exist in a species because it happens as a sort of an error during PCR where the DNA gets joined. Okay, so those are chimera molecules. So the DNA from the red butterfly and the blue butterfly join together during PCR to make what looks like a new species, but is not. Okay, chimera. And it's difficult to distinguish contamination and false positives and negatives. So sometimes, for example, you have 3 species, but you maybe get 4 sequences. And it's hard to tell. So you can see there's a, what rather, naranja, right? Is that the fruit or is that the color, or both? Both? Yeah. Daniela taught me that. Naranja. You don't know if that, because when you look, you don't know this. All you see is this, right? So you don't know if you have an orange species, or there's some contamination, or it's a sequencing error, or maybe it's a chimera, and you don't know, right? So that's a challenge. How do you make sure you maintain the real samples in metabar coding? Any questions so far? Okay. And you might hear the term eDNA or so as well, and I think you can think of eDNA as the eDNA metabar coding, right? So it's all it means is you have multiples, you have DNA from many things that you're barcoding at the same time, okay? Whether it's eDNA, which is usually just small molecules that are degraded from the environment, or full organisms DNA. It's the same as long as you put it all together. Okay, so when you're clustering reads at step 1, you get many what we call amplicon sequence variants, or ASV. And so if you have one species, you can have variation in the sequence for 2 regions, right? So so if you have, say you have 100 individuals of the same butterfly species in a bottle, you don't expect all those sequences to be the same. So why? There's 2 main reasons. Why might you get variation? See? The same species, but maybe different populations, for example. So you can have real genetic variation, right? So that's important. You can have real genetic variation. And second, this one's maybe less not biological, but more something that happens as a technical matter. Sequencing errors, exactly. Or maybe chimeras, exactly. So you can have real biological variation and sequencing error. Okay, that's important. And so we want to know what species are in the sample. Why do we cluster them instead of just keeping every unique sequence? Well, the reason is if you keep every unique sequence, you'll have so many sequencing errors, right? Every single time you have 1 or 2 differences, that would be a new thing. You can't really tell that apart from real biological variation, right? If you have an A and a T in one individual and a C and a G in another, you can't tell if that's a different individual or if that's error. So you just cluster that together, okay? And so when you're doing clustering, read clustering, this is something that we'll work on a little bit. If we get Ubuntu working, or I can show you on my computer. The key parameter that we use, there are several, but the key parameter is the clustering threshold, okay? And I'll explain what that means. So, let's imagine we have 3 species, real species. We have a red species with 3 individuals, light red, a little bit darker, and then dark. Morning. The blue species, we have 3 different colors. And we have species 3, the black species, 3 different colors, red, gray, light, darker gray, and black. The DNA looks like this, okay? What we want to recover in a good analysis, it would say we have 3 species. If we had too high of a threshold, meaning we said the species have to be maybe almost identical, how many species do you think we'd see? We have a very high threshold. Would we see it's too high? Can I repeat? Yes. Yeah. So if we used a very, very high threshold, maybe let's say 99%, right? And we have these DNA molecules here. So 9 different types of DNA molecules, and our threshold was too high. How many species or how many clusters do you think we might get? Who thinks
5.
9? 9?
No. 5.
6? I get 6.
1, 2, 3.
We would get more than 3 is kind of what we would, is kind of the answer. You might get 9, you might get 6, because what we're saying is, we might say that this is different, this is too different from this is too different from that, this is too different from that, right? And so if we use a very high threshold, we are separating things that shouldn't be separated, okay? Okay, what if it was too low? 90%. How many species would we, how many clusters would we think we have?
5.
It's too low, remember, so there's 3 species, but the threshold is too low. What do we get? 3 would be
3,
perfect.
5.
If we had too low, we just get 1. Okay? So if our cluster is too low, we keep everything that's pretty different. We would get all of them saying the same. But if we use the right threshold for this clustering distance, that's when we get the right one, right? So we have the threshold that's high enough that it's not lumping everything together, but not so high that every single sequence is being treated as a different species. Okay, so that's really important to sort of get right. There is no correct answer. You just have to do your best and learn from real data. Typically we use like 95, 96% for metabar. Okay, and so one of the things that we'll do, maybe you'll do on your computers, there'll be a chance to do this as an advanced lecture, we can figure it out, how to work on these computers, is using the bioinformatics commands that we use to cluster a FASTA file, meaning a group of sequences, with different thresholds, and we'll look to see what this looks like. And so what we'll do is we'll use, so we'll do that, and then we'll use Unix to count how many sequences remain in the output file, okay? So that's a preview of what we'll do this afternoon. So we'll do the clustering of reads. Okay, so next is the chimera screen, right? So we've just talked about the first problem of how you cluster your reads. Next is chimera screening. So when you have chimeras happen, because you amplify DNA from different species at the same time, in the same reaction, they can join, right? So you can see here, we have the blue species, that's their DNA, that's their barcode. Red species, that's their DNA, that's their barcode. And the red and blue species, that's a chimera, right? So based on what I'm showing you here, what sort of logic could you use to tell if something's a chimera? And remember, you wouldn't see that as red and blue, you would just see A's, T's, C's, and G's, right? So what could you do to see if you have, DNA 1, DNA 2, and DNA 3, because that's kind of what it would look like, right? You don't know it's a chimera. What could you do with these 3 to see if one is a chimera? And I'll give you a hint. You have all the information you need there. Stop codons? Not necessarily. The stop codon could be introduced, but it might not, because you don't have any stop codons in the real sequence. Size of sequence? Probably not. It might, but they could be just the same length. You said? Yeah, you can compare them, right? You can compare whether parts of them overlap, exactly. Yeah. So what you do to screen for chimeras is you see if any molecules can be made up by other molecules, okay? So what you do, if we have different ones, we call them parent 1, parent 2, and then the one we're checking, query, okay? So if we had this chimera, we check all pairs. We check here, we can see no part of that query matches this, okay? So that molecule, we would say from this, it's not a chimera of those. But, okay, and there's another one. This molecule, what we can see is that when we check against parent 1 and parent 2, yes, we see this part in there, but this part in the box, that's unique to the query, right? So chimeras won't have any part that's unique to them. So we would say, because this query has something unique, it's not a chimera, at least of those two parents. But, in this case, this query molecule, there's nothing unique, right? Part of it is from here, and part of it is from here, right? So we can say that is likely a chimera, right? If we do that for every pair, we check every trio, every query, every molecule is a query against every pair of parents, then we can see which ones are chimeras and remove the ones where they're made up of potential parents. That make sense? So we go through every combination of 3 in every configuration, and we say, okay, is this possibly a chimera? Yeah, and then we can remove it. Okay, we're gonna do that. And so what we'll do is go through how you would do a chimera screen on the command line. So I have some files that have some real molecules and have some chimeras, and we'll use a program, which is in the program research called UKIME de novo. Not to cluster a fast with different thresholds, to screen for chimeras. That's a mistake. And then we'll look how many chimeras were in there, okay? Okay.
If there's no skipping ahead, Ken, sorry, I'm not having any luck with this. We don't have administrator passwords for these computers, do we? It's
No.
saying no, research is not installed, we need to use sudo to install
We can't
research.
install.
Yeah.
Okay. So we'll just have to probably do it on here. Maybe I can set up some virtual machine or something. I'll figure it out. See what I can do in 10 minutes. Okay. The last thing that's on the Ubuntu.
This is on Ubuntu.
Do you want to see if you can install WSL? Windows on Windows?
Windows.
Yeah. See if you can install Windows Subsystem for Linux, and then vSearch might be in there. We'll see. Okay. Okay. So the last thing is we want to separate the real ASVs or clusters from ones that are either contamination or mistakes. And so you remember this happens a lot in PCR. So you have the plate, right? Let's just show like the top corner. We have like A, B, C, and then O1, O2, O3. Then we have the wells. So that's what the plates look like, right? And you have different samples here. The blue sample. Well, when you're doing PCR, you're dealing with liquid, and you're dealing with heating it up and cooling it down. And sometimes you get like little tiny splashes that go away, right? So you get really small little splashes or something like this. Let's say you had 1,000 copies in here, in the A1 of the blue. The splash would not give you 1,000, it would give you maybe 1 or 2, much less, right? Most of it is in here. So what we can sort of say is that if something is real, it's probably represented by lots of copies. And if it's just a splash or some contamination in the air, or just, stuff comes in from the air, it's what happens, it's probably a few copies, right? So what we do generally is we look at our sample, and actually if you look, most things will just have 1 or 2 copies, right? So we say those are probably mistakes, those are probably errors, and the ones with lots of copies are probably good. So let's say we had in our sample to look like this, okay? We had, in this case, 6 real species, and we have 3 errors, okay? And we have, this is how many reads. So for this molecule from species 1, we have 167 reads. That's how many times we see that that ASV. This one 120, this one 20, this one 6, but it's real, but 6. This one 1,400, this one 353. And then this one, errors 1, 2, and 1. Okay? So you have many copies, but variable, but many, of the real, and a few of the errors. Okay? Usually, yeah, so yes. If you have no filtering, if you just say, okay, this is my data set, 1/3 of your data is an error. Okay? 1 in 3. That's not good. So you'll say we have 9 species. It's a mistake. You have 6, right? If you do too much filtering, so it's not showing up so well, but let's just say, you can maybe see if I do this. See those ones that are changing a little bit? Which I changed the number. If you've required 20 reads, say, then you lose this species, this one, and those 3. So, if you require 20 reads, then you accidentally delete from your data 2 real species, okay? So that would say, we call that a false negative. The species is there, but you don't see it because you filtered it out, right? So you want to minimize false negatives. So what you want to do is minimize false negatives and false positives. Those initial, if you include these in there, which might be real species, but they're not in the sample, they maybe just come in as contamination, that's a false positive. You find the species when it's not there, okay? So for this data set, and many real data sets, 5 reads would actually give you the right amount, right? It's getting rid of the errors, but it's keeping the real ones. And so the trick is to find the minimum number of copies of reads that allows you to get the best data set without having mistakes. Okay, and so one thing we'll do this afternoon, some of these actually you can do in PowerShell. So this one we can do. We can remove sequences that have, we can do the read filter. We can say we'll drop out sequences with too few reads, right? And so we can sort of say, okay, remove ones with 20 reads or more, remove ones with 5 reads or less, and you can see how many are in there. And so we'll use that. And that one, you'll use a new command. Maybe some of you have used called awk, and you can figure it out with ChatGPT. So for me, I'm not the best Unix programmer. I'm okay, but I'm not the best. I use ChatGPT for everything. I used to be better, and I've kind of got worse, but ChatGPT is really good for this. Okay, so any questions about, before we move on, any questions about some of these basic bioinformatics approaches we use with metabar coding? So that's read clustering into ASVs, chimera, screening, and filtering based on copy number. Yeah, Danielle?
How do you recognize that in the previous example that an n equals 6 is real species?
So the question is, how do you know which one's a real species versus not? The answer is you don't. You just need to do your best to be confident. These types of answers are what we do. Is we develop, we calibrate different results by, we would do metabar coding, and we would barcode every single specimen in the same sample to sort of learn about what we can expect. And so Robert and I have been working on a project where we have 10 samples from Australia, and we metabar coded each one. Robin and his team has been doing that, and then the biodiversity factory at Biodiversity Center for Biodiversity and Genomics has barcoded every single specimen, and then so we know what's in there, and so we can start to say, okay, we see 20 reads, we see 10 reads, which ones are false positives, which ones are false negatives. And so in that case, in that data set, actually 20 reads is the right amount. That gets you the right data set. So I think you need the big core facilities like us to do these really expensive analyses that do the barcoding to sort of generate the expectations, and then other people can sort of apply our principles. Yeah. Yeah. But yeah, you have to calibrate based on data where you know what's in there. Yeah. Any other questions about basic metabar coding, bioinformatics, filtering, screening? Okay. So, I don't know if this will work because Bold has still been having some problems, but we're going to try some things. So originally, metabar coding used short read sequences. So, what this would mean is this is the whole barcode right here, the whole thing, but all you would see is a part of it because on the old sequencing machines, you might have heard of Illumina sequencing that we've used, the size of the DNA molecules that they sequence are shorter. Often they're 150 base pairs or 300 base pairs. So you can't, in a single molecule, see the whole thing. And so sometimes you have even shorter, right? So this is the whole barcode you might get, it might be 100 molecules. Any luck over there, Robin? On the Windows stuff?
Oh, no, sorry, it's WSL's not installed, and I tried installing it, and again, it needs
Wait,
a
is it a share?
password. I'll do that.
Okay. And so, yeah, if you have any input also, Robin, you can feel free to chime in.
Yeah, sure. I was just going to say, I mean, you said old sequencing platforms, not even old necessarily.
Yes, not old.
Illumina, I mean, they are still used. A lot of applications, all you need is short reads. And these platforms give you enormously high read depth very cheaply. That's why they're still done that way. So that's, in many ways, that's the reason metabar coding was. It was sort of built around these short reads because initially those were the only kind of platforms where you could get the kind of read depths, the numbers of reads that you need to implement.
Yeah. Yeah. So that's correct. People still use it. It was the way they started with short reads, and they're still doing it. That's the way actually CBG primarily does the metabar coding.
So metabar coding with long reads is a relatively new thing. I think we were kind of the pioneers of it in many ways because it's a newer platforms like Nanopore. It's only relatively recently that Nanopore has gotten good enough that it can give you the kind of read depth and the kind of sequence quality that you need to do metabar coding. So we can do full length CO1 at metabar coding scale.
Yeah.
But that's kind of a recent thing.
Yeah. So the full length, by quiz, I don't mean quiz, I mean discussion, not quiz. The full length CoA or CO1 barcode is 658 base pairs. If we sequence, this is sort of based on what Robin was saying. If our molecules from our sequencer are 300 base pairs long, right? So here's the molecule. We get reads off the sequencer that are about, so that's the CO1. The sequencer length molecule from Illumina, which we call the amplicon of the sequence, or the read, is like this long, 300, 658. Why, if you have a 300 base pair amplicon, why would we maybe only get 150 base pairs of usable barcode data? So why is it the case that a 300 base pair amplicon doesn't have 300 bases of barcode, has 300, or has less than that, maybe 150?
We are only sequencing the bilateral origins, of that name.
We're only sequencing the variable regions. Not so. We want to sequence the whole thing. Certainly, there's more variation some places than other. That's not quite why. So what else? If you sequence 300 base pairs, what are you sequencing?
Think about the technicalities of how you do sequencing, like what you actually have to do in order to get that thing that you're sequencing.
Simona? No? Prime? I heard someone say primers.
Primers in the right area.
Exactly, yes. So, when you sequence a 300 base pair amplicon, you're sequencing, there we go. You have your barcode in the middle, here. Then, on the outside, you have primers, You have different things, spacers, sequencing primers, adapters, indexing tag, right? So you put different molecules on the outside of the barcode you want to sequence, and that you sequence those. You need to sequence the index, like the UVs, right, to know to do the demultiplex stuff. But that takes up your sequencing space. So short reads, you kind of lose a fixed amount of your sequence to, we might call it logistical requirements of sequencing, right? Which is, it is what it is. It's not great, but even with the 300 base pair or longer amplicon, you still only get a short amount of barcode DNA. And so at the Center for Biodiversity Genomics and Innovation team, and this is something that Robin has really worked on a lot, we're developing protocols to do long read metabarcoding, and that's exclusively what our team does now in terms of the innovation team. We don't do Illumina barcoding, we only are using nanopore.
And just to mention that what Ken said is still true of the nanopore sequencing we're doing. It's just less important because with nanopore read length is not really an issue. Like when we sequence the 658 barcode region, the actual thing that gets read by the sequencer is more like probably 700 to 750 base pairs, because you have to include the primers, you have to include the unis, it includes those adapters which are part of the OMT sequencing that have the motor protein. So that is all extra sequence that gets added on. Our bioinformatics just essentially filters all of that out, trims it out, and we get the sequences. So you won't see that in the sequences that we finally get, but bear in mind that they are always there as part of the reads that the Oxford Nanopore device will generate.
Yeah. And so this is what we switch to now, because the Oxford Nanopore generates reads can be tens of thousands of bases long, right? Very, very long. And so we can easily sequence the whole thing, plus all the additional stuff that Robin was saying on the outside. Okay, so I developed this little exercise that we can do together. Let's see if Bold works. On, I think it was yesterday or Monday, yesterday. Bold was not working so well. Yeah, thanks. So we're going to try to use Bold and barcode ID to examine the consequences of doing barcoding with short molecules versus long ones. For now, let me start. So don't do this, just because if Bold is slow, then having many people use it would be not good. So let me start, and then we can try it again. So what we're going to do is we're going to use the Bold data portal, and we're going to find a species which is in the monarch butterfly genus in Latin America, Danaeus Montezuma. Okay, so let's go here. I am not online. I developed this when I thought there would be internet, so I need to switch the monitor. I don't have Wi-Fi. Maybe I've told you that already. Okay, that's okay. Are we on Ubuntu? It'll be fine.
This is still on Windows.
That's okay.
Yeah,
so
yeah. I was asking if you want to switch the HDMI
Oh, yeah.
to the PC.
Yeah. Let's see if it works. Okay, good.
Do you want this light off?
Yeah, let's turn the light off. Okay, so don't do this yet. If we can do it, if it works, I'll make this bigger. Okay, looks like it's going pretty slow, so it might not work again. So again, let's see if it works, but the Bold data portal is how you generally quickly search the barcode database to get information you need. Let's see if it works. So if I want a species, I would type, okay, looking good, Danaeus Montezuma, that's a taxon. Search. Let's see. Pretty slow, but it might work. No one do this. Don't do it. Usually it's fast. This week, slow. Let's give it a minute. I think maybe we just can't use bold to teach. Let's give it one more minute. Let's see if it works. Okay, I think it's not gonna work. I don't know why. No say. Let's try a different browser. Let's try Microsoft Edge. Close. Now I want to do this. Okay. So we go data, portal. Don't do it. Launch portal. Okay, never mind. Doesn't work right now. Someone will get in trouble. Okay, that's gonna put us ahead of schedule. Okay, well, normally bold works. This week, it's not working. Okay? Good. Skip, skip. Maybe we'll try later. Skip, skip, and skip, and skip. Okay. So that would have taught you something useful, but I hope we can teach you later. I need to write to the team and see if they can kick everybody else off bold and let us work. You know, I'm gonna write to Paul. Maybe Robin, do you have any internet? You don't, right?
No,
Could you phone, not phone, text or email Paul and just say like,
Yeah.
can you see if you can restrict bold to only Peru for like an hour or something? See if they can make it work, because I would like to do that exercise because that's pretty important.
Yeah.
So, okay. Sorry about that. Impossible to predict that bold wouldn't work for us, but it is what it is. Okay, so we at CBG, and me, Robin, our colleague Sean, are developing something that we're calling Maple, which is the metabar coding analysis pipeline for lineage evaluation. This works with long read metabar coding data to do everything that we said. So it makes sure the molecules the right length, demultiplexes them, does the ASV clustering, looks for pseudogenes, pneumites, chimeras, and also it identifies taxa using our new library. And so this works. This is as fast and as good as possible. The best way that we know how to do metabar coding. And the whole pipeline, especially those first parts, the computers that we have here are not quite powerful enough to do, but we'll do parts of it on some example data. And one of the things, I guess since vSearch is not working, we might have a hard time doing this. What I can do with you is show you how we do the taxon matching with vSearch on our computers with reference libraries, which is very easy now. Another thing that we're building is something that we're calling RAMS, which is sort of like bold but for metabar coding, which is the registry of amplicons or ASVs from metabar coding studies. So the data from the output from Maple gets uploaded onto RAMS, and we're developing this now. Okay, and so one thing, just a little discussion point. When we take the metabar coding results from Maple, we tell the user what the taxonomy of the ASV is, but we don't take that information and put it online and store it ourselves. Why do you think taking the taxonomy that's assigned when you run a metabar coding sample and putting that online might not be a good idea? Let's think about that. So let's say I have a sequence, it tells me it's in the genus, the butterfly genus we looked at earlier, Danaeus, but there's another species. Why do you think that might not be a good idea to put online? Maybe it's difficult. It might have been more obvious if we did the exercise. The reason I'll just say is because the taxonomy changes all the time, right? As you get more information about the molecules and things like this, you update the taxonomy all the time. So even a week or 2 weeks after you do an analysis on metabar coding, it might be new. Your taxonomy that you got from the analysis might be wrong. And so what we do is, using the data online, we just update the taxonomy within the database rather than use the taxonomy that was initially assigned. Okay? I also don't think you're going to be able to do this shell script, but we can sort of show you the data structure that we put on RAMs. Okay, so just a little bit on the future of metabar coding. So, and this is increasingly true, almost all sequences of COI or CO1 are going to be from metabar coding analysis. Very few, relatively speaking, just because it's so much more expensive, will be from barcoding. More and more barcoding will be metabar coding. I would say even now, Robin, probably most CO1 sequences that are generated are from metabar coding, probably. But certainly in 5 years, that's going to be probably almost all. I think also, as the long-read sequencing technology gets better, metabar coding will keep using the long-read sequencers like ONT, or PacBio. And because of this, we need to start developing tools and technologies that can link the different sequences across studies. Just like bins do for barcodes, we need to start having systems that can link different metabar coding results across those studies. Okay, so any questions about metabar coding? Well, if we can, we'll try to come back and do that exercise that I had planned later. But because we can't go on hold right now, it's not working. So yeah, any discussion, questions about metabar coding, bioinformatics? Yeah, Emma?
Is in the case of the eDNA, for example, is better use nanopore sequencing, right?
So, question is, in the case of eDNA, environmental DNA, is it better to use nanopore sequencers? I think it depends a little bit on the average molecule size you'll get from your eDNA. So, from like air eDNA, how big? Do you know how big the molecules are? Maybe do you know, Robin?
Short.
Short.
Probably like below 100 base pairs.
Yeah. So what happens when eDNA? I'll just explain. Okay, the audio does anymore.
Yeah, DNA that is not inside an organism, if you like, that's not recently alive, very quickly gets broken up into short fragments.
Yeah,
That's
so we have
why
a little
DNA is
fish.
short.
A little fish. How do you say it? Pescado is fish meat. What do you say? Fish? How do you say it?
Pes, pes.
Pes?
Yes.
Pes. Fish? Pes? Okay. So this fish has DNA. Barcode 658BP. Maybe it goes to the bathroom. Then you right away might have 350 base pair because the DNA just gets degraded by the environment, right? It's getting out the side. Then, with time, maybe 1 day, then it's shorter. Then it's 100 BP because it's not being maintained by your organism. It's not being repaired, it's not being maintained. So over time, so this would be DNA, this is eDNA, or environmental DNA. And so, yeah, when it's in the air, if it's in the water, it gets shorter and shorter and shorter. Air especially gets short, just because it has to float around, so it has to be small. And so, if it's 100 base pairs, and the Illumina reads are 300, it's probably okay to use Illumina. Probably better. Because I think Illumina reads, just because they're designed to be short, they're more accurate, they're cheap to generate. But sometimes you can find reads that are a bit longer in water. You can find like 300 base pair amplicons. In that case, you probably would want to use nanopore, right? So if you're targeting short, Illumina is okay. If you're targeting long, then nanopore is better. Robin, do you have anything you want to add?
Yeah, no, just, it sort of depends on how much sequencing you want to do as well, because nanopore is, because the flow cells are cheap, the sequencer is cheap. If your project is relatively small, that might be your better option as well. Illumina, like, I don't have numbers, but I think they are quite expensive per run.
Yeah,
You do
yeah.
get many, tens of millions of reads per run, so it's cheap per read, and the reads are very accurate.
Yeah.
So,
Yeah.
advantages and disadvantages either.
Yeah. Any other questions? Okay, so now we will do some more advanced R exercises. And we'll try to come back to this later if it works.
Vamos a poder hacerla.
Por el bol.
This is up in check.
Pero si nos da, me imagino que se buscará
Thanks.
el pasta, ¿no? Pero si nos da el pasta por correo.
Okay, and let me make sure I have this.
¿Has tenido tu laptop?
No,
Porque también digo que se puede poner.
sí,
Okay.
la dejé, pero
So, for this exercise, I'll make it bigger. We're going to use the script 3 intermediate, okay? So that's what we'll start with. And just to start, everybody please, this should work, but write the following and run each line. Library, tidyverse, run, okay? And library, gapminder, run. So run both. And yeah, keep the blue stickers and orange stickers on the computer for today, just so you know. You don't. That doesn't matter for this one, but when we do what's called bold connect R, when we're using R to connect to bold, then we need to use the right computers.
me lo voy a ver acá porque
Who's using personal computer? Fernando, you are? Anybody else? Okay, so Fernando, Windows? Okay. Can you type this, Fernando? Let me see. Do you have DevTools installed, do you know? So maybe not, so type this. No one else do this. So say, Fernando only. Install, packages, and then quotes, DevTools, run, then, you always protect that. Then, in the script number 4, there's this line right here. It says DevTools install underscore GitHub. Run that. It has a comma, so you have to delete that. So go into number 4 and run that line. And then I'll check in. Okay? Everybody's done this? Nobody's done this except for Fernando. Okay. That'd be okay. Who's ready for some more advanced R? Everyone's good? Okay. And again, you'll use all these techniques, I hope, when you're doing your project looking at barcoding data, metabarcoding data. Okay? So, what we'll do is explore some even more advanced data wrangling functions in tidyverse. We'll review a little bit of what we did yesterday with pipe as well, and there's other things. Select, filter, mutate, summarize, group by. We'll do the very basics of some more advanced techniques like loops and functions. And then at the end, we'll use R to perform some basic bioinformatics, and we'll work a little bit on the command line. We'll see, I might have to do it on my computer. I'm not sure we can do it on these computers actually in the end. Okay. So the first things we'll do, is that it? is what's called pivoting. Okay, pivoting data. I do this all the time, every day. Because sometimes the way that you write data in the field or you get data from other people is not the way that you need it to be. Or the way that you have data one time is you need to change it a little bit to be better for a certain analysis. And so knowing what you can do with pivoting, sort of I would say making those neurons in your brain, developing those neurons, makes you a better data scientist. It'll just make you better at every single bioinformatics, everything. Knowing about piping, knowing about all this, just having your brain sort of update that software firmware is good. Okay? And sometimes like when I was in doing my PhD in grad school, I did a lot of experiments. And because I was good at the data science and data wrangling, I could design my data sheets to be collected very efficiently, and I didn't have to record any redundant information. I could enter data very fast, and then do the things on my computer with it that I needed to do later. Because I was confident I could do that, I was better at doing field work, right? So being good at data can really help you in a lot of ways. Okay, so the first thing we'll do is what's called pivot longer. Okay? So what this does is it makes your data set, used to be called gather, now it's called pivot longer. Makes your data set long. So it takes it from being wide and then it kind of pulls it to being long by collecting columns into a row. And so what we'll do is use pivot longer on the gapminder data. Okay, and I'll go back, I'll show you what this does, but essentially the way this works is it takes data that looks like this. Okay, so you have one sort of key column here, and you have a bunch of columns that you want to make into a row. And so you take these column names and you make them values in a new column called year, and we have another column which is the numbers that were in there. Okay, so you sort of see it goes from wide, you pivot to long, right? So the data is the exact same, it's just formatted differently. So let's try this in R. So if you go down in exercise 3, we have some code here that will do that. So if you see here, the gapminder data, if you look at it, has 6 columns and 1,704 rows. And so what we want to do here is make it longer. So what we do, the term is you specify the data. We're not doing it in a pipe. We're going to take these columns, and so I'm using the colon to connect them. So this means life expectancy, and then the colon means until or to GDP per capita. So that's those 3, okay? So you can connect them. Then we want to know what our new column is going to be called. So if you go back to here, the new column is called year, right? That's what those are. And then the values, what are you going to call that? And here they call it cases. This is maybe the number of cases of a certain disease. We're calling it just value, okay? So what we do, you can run this. Then you can look at what it looks like. And you can see now there's only 5 columns. Our new column, the name is metric, life expectancy, population, GDP per capita, and all those numbers are in the value column. Okay? So we made this data set longer. It originally had 1,700, now it has rows, now it has 5,012. Okay? And we might want to do that because you might want to make a plot that looks something like this. If you run the next line from 22, sorry, it's like kind of hard to see. What can I do? How are you doing? Now this one? Yeah. So now you can make a plot that looks like this, but you couldn't. So you can sort of say, how do these metrics across the different continents vary by year? So you can sort of see life expectancy in Africa went up, then has mostly stayed the same. Most other continents it's going up. Faster in Oceania than Europe. Oceania has the highest. How has population changed? Some Asian countries really high, India, China, or China, India. GDP per capita, how has that changed? You see some started really high, had the highest ever, then went back down. Right, so if your data wasn't in that long format, you couldn't make a plot like this. Okay? And also what you can see is I used a command that made the scale of the y-axis different for every graph. So I made this one go up to 90,000, this one go up to 80, this one goes up to like a billion or more. Okay? 100 million or yeah, hundreds of millions. So that's why you can pivot the data. Okay, go back to lecture. Okay. Augusto, ¿está bien?
¿Pudiste hacer? Bacán.
Okay, okay. Is the functions are working?
Yo entiendo que esto lo estamos haciendo porque, o sea, estos son datos poblacionales, ¿no? Yo lo entiendo así. Pero esto se puede hacer también para ver diversidad, abundancia de especies, ¿no? Con las secuencias, ¿no? O sea, sí, nos está enseñando más o menos la ruta, el camino, cómo hacerlo para poder visualizar.
Y también para intentarlo así como
esperanza de vida, el PBI, la población, ¿no? Cómo,
para entenderlo así.
claro, comparar estos datos importantes.
Okay. So, the opposite of making data long is to make it wide. Okay? Sometimes you want to make it wide, sometimes you want to make it do long. Sometimes you want to go wide and long in the same way. I do this all the time. So, this one is called pivot wider. It used to be called spread, now it's called pivot wider. So it makes more columns. Okay? So the way this works is you take your data. I can do a select just to keep certain columns. Then you specify which variable you want your new column to be. You want to spread one, or you can do more, but one is typical. You spread that one column across where you want to get the values from, and then you can do other things to change the name, which I'll show you now. Okay, so basically the way this works, it's the exact opposite. So you can pivot and then go exactly back the way you were. Okay, you can pivot along Y, pivot along Y. No new data is made or anything like this. So here, this is long. You're pivoting Y. You're taking the names from the year column, so those become your column names. Okay, there, and the values from the columns and cases. And so those values in the purple go in here. Okay, so they're a little bit different than before. Here, names to, values to. You pivot Y, names from, values from. Okay? Once you do this, once you make these sort of brain connections, this will be like speaking Spanish. Okay? It's like, for me, no problem. But you just have to do it and get used to it. So let's practice. Okay. So we have Gapminder here. Let me pull this up. So this is the raw data. Okay, 1700 by 6. We do a select because we don't want too much. So we're going to spread life expectancy by year. So we just keep. You don't have to do. I'm just doing this robot to show you. So now we've just selected the data. To keep country, continent, year, and life expectancy. Okay. Then what we want is you want year to be the columns and life expectancy to be the values. So what we do is pivot wider, names from year, values from life expectancy, and then this is just something I did because if column names are numbers in R, that's weird. So I added a prefix. So it'll say life expectancy to prefix. Life expectancy underscore 2002. Life expectancy underscore 1997. You don't have to, but numbers as column names in R is weird. So if you run, and you look, I call it gapminder Y. Here's what it looks like. You can see here, looks different. Life expectancy 1952, life expectancy 1957, life expectancy 1962, right? So this is common. If you're collecting, say your own data, you're making repeated observations on the same individual, right? Say you want to say how big was this fly when it was 1 day old? How big was this fly when it was 2 days old? You'd want to collect data in this format, right? Because this is much easier, right? You can just follow the same fly this way, or if you're wanting to measure the temperature at the same environment over time, you want to collect data this way. But when you want to analyze data, you might want it the other way, right? So being able to go between these two ways of the data is really important. Okay, so now we've made this Y. Okay, so we're going to do some exercises as a group, and I'll put the other, we're going to have you pivot the data, pivot some one way, and then do the other. We're going to have you do both pivot wide, pivot long. So can everybody put, oh, you don't have them. Those guys, they came and took the colors. Okay, can you guys, jeez, can you pass these around? Actually, yeah, so just take one of each.
We are getting hugely variable response.
I'll come back. I'll try when you're there. Okay, so I just want to show you the data we're going to work with. Then we'll start the exercise. It'll take some time. This is challenging, but it's okay. You need to learn. So what we're going to have. So you need to run the command data billboard. That loads data into the memory. If you're interested, part of the reason R is slow is it always loads data into memory, and that's really slow. Okay. What this looks like is like this. Okay. Artist, so this is the billboard like music charts. Artist, song, when it entered, and its position during different weeks. Okay? So every column is a different week. Week 1, week 2. Who thinks this data is long? Who thinks this data is wide? Okay, you have to answer one. Is this data long or wide?
Right.
Wide, right? So you have view billboard. Let's see. So the data looks like this, very wide. Okay, that's what it looks like. You can see very wide, 79 columns, right? So what we want to do is take this data that's very wide and make it long. Okay. So, I'll pull this back up. I'll pull up the helpful chart. Call the new data. Wait, let's see if I have an exercise here. Okay, so we're going to take the billboard data and make it long. Call it this. So, I'll give you the starting. So, billboard underscore long. Then we can do the pipe. We can do this. We'll do pipe. Billboard. Yeah, do it with the pipe if you can. So then pipe. Next line. Pivot longer. And then it's up to you to figure out what goes in here, okay? So everybody, if you have the yellow, put your yellow on your computer. So go back here. Okay, so everybody work on pivoting that longer. And when you're done, let me yeah, and I will pull this back up because this will be helpful.
So I may go see Jorge now and set up the afternoon stuff.
Yeah, that's good, yeah.
Keyboards, longer.
Yeah, I'm gonna have to improvise a bit with the metabar coding stuff, but that's okay.
Data longer.
And switch to green, so yeah, please put the yellow on your computer. Switch to green when you got it. Where are the sticky? Take the board? No. Take,
Quiero que la ponga ahí.
so switch to green when you have it. So when you get it, switch to green.
Ajá. Sí, pero bueno. Tú.
Red if you're stuck. I'll go to the bathroom, be back. I don't have it on my computer, so no cheating. You can't find it.
¿Lo corriste? No, pero si estás viendo el informe de diferentes tiempos, ¿no? Que estamos viendo una data de países.
Claro, no existe.
Era de semanas o algo así. Era de semanas o algo así. Columnas.
Porque en el otro era ya diferente. Era esta y esta. Y se cambiaba metric y volumen. De acá a acá.
Por eso, pues semanas.
Ajá, le pongo una week 46.
Okay, I'll give a hint. Who wants a hint?
Green.
Or who wants no hints? Okay, I got it green. Who wants a hint? Okay, I'll give you. I have to just double check how it's spelled.
Week. 41.
7.
Ujú. Ley.
¿Qué más?
Let's make sure Bella has it
Wait. No, doble. Doble E. Sí. Sí, pero sin la. Ajá.
Okay, so I'll just tell you a little hint.
El valor.
First thing to do,
Ya, ya.
week 1, and then use the colon, then the rest you figure out. That's the first thing.
Sí me dio, pero solamente hasta las 47.
Valerie, now it's your job to help
46.
Ajá.
the other students. Valerie, you can help the other. You keep the green.
Arreglar.
You can help other people if you want.
Sí, ya.
Yeah, good, good.
1.
76. Bueno.
Tengo duda de eso. Estuvo y varios.
¿Le llamamos a.?
Sí, lo que. ¿Qué es esto?
A ver, ajá, abre ahí.
Voy a borrar esto ya. Esta era. ¿Cuál será la nueva? Esta me imagino, ¿no? Sí.
No, no, eso es lo de los países.
Ay, ay, ay. It's easy to trip in this room. Okay. Let me check. Does it work? Let's see. Yeah, run. Did you run it? Oh yeah, yeah. Está bien. Yeah.
Okay.
Good.
Okay.
Verde.
Verde.
Verde. You have it, Stephanie?
Hay que ayudarle ya sin
Is it working?
O sea,
You want to pivot? You want to see
hasta el
that? First
76.
thing.
Sí, nosotros cuando
You want
nos
to
hasta 47, después nos lanzó.
Hasta el 76.
Ahí nos enlaza. Hemos copiado de acá.
And remember, if you want to look at the whole data set, if you need to browse it, you can use you can use view. I'll show you. View. So if you just want to have a look at the data, you can use capital V, IEW, Bill, or
Beautiful.
yeah, that's good.
Ahí está. Beautiful.
No. Can you click the billboard? Right. Oh, that's backwards. Yeah, it's backwards. No, it's pointing this way. It should point that way. See if you can figure it out. Do you know? It's, I'll write on the board. There's other ways too, but yeah. So you can change the last week that you wrote to that. Right, put a period. Yeah. Not quite. So, want to see the email? You get